All right, guys, we're going to go ahead and get started. First of all, I want to thank you guys. A lot of you have provided a lot of contribution to this project, and honestly, I don't think it would have come out as good without all of your help. So thank you so much for everybody who has contributed to this presentation. No disclosures for myself, for Dr. Bolag. He is the medical director for the MBTC blood component testing laboratory. And we also used some AI in the production of some of the images that you'll be seeing today. So imagine this. You've finally been promoted to the lab director at your hospital's blood bank. You've only served a few weeks in your new role, and you've barely had a chance to get comfortable when the FDA arrives with badge in hand. They hold an open meeting with your hospital's leadership. They provide them with a Form 482. The blood bank is on that form. You're being investigated. So why worry? Because blood and blood components are intended to treat and prevent disease. They are classified as drugs under the Food, Drug, and Cosmetic Act. The Code of Federal Regulations requires that your blood establishment be registered annually with the FDA and list the products that you manufacture. More so, you are required to abide by the current good manufacturing practice standards as established by the CFR. And unlike the deemed organizations like CAP and ABB, the FDA carries the force of law. They can and will shut you down. Before your inspection, you receive some helpful tips. Be responsive, cooperative, and transparent. Any defensive behavior, delay in documentation, or inconsistent answers will erode trust. Never alter documents under inspection or attempt to recreate missing records. If an issue is identified, acknowledge it professionally and follow up accordingly. Designate a subject matter expert that can represent the facility during inspection and address any questions throughout an escorted walkthrough. Naturally, you're nervous, but you remain calm and composed. You are cooperative and transparent as the FDA reviews your policies, SOPs, and quality control and assurance documents. They review your donor contact information and the last 10 years of product and donor records. You escort them on a walkthrough of your facility while they apply a systems-based approach to their inspection. You are responsive. You diligently monitor and note any observations to report back to your staff for follow-up later. You promptly and truthfully answer any and all of their questions. You even take time to educate the inspectors on your equipment and your processes. At the end of a long day, the lead inspector thanks you for your cooperation, but she has concerns, a lot of them. She hands you a Form 483, but this is just the beginning. A typical FDA inspection lasts just 3 to 5 days, but over the next 6 weeks, that 483 has grown over to 10 pages long. You and your quality management team are quick to write a strong and detailed response within 15 days. You know that an unsatisfactory or delayed corrective and preventative action plan will result in further escalation to a warning letter and possibly further legal action. So you prepare a strong response. You address each point separately using root cause analysis tools such as the fishbone diagram or the 5 whys method. You create detailed corrective and preventative action plans that include clear timelines. You provide evidence of your CAPA implementation, such as updated policies and SOPs, training records, and improved document management using electronic record services. You demonstrate your system-wide improvements that you've made, such as modifications to your governance system or quality management team, equipment and software updates, and modifications to staff and workplace environment. The FDA reviews your response and they're pleased with your team's responsiveness and accountability. They've approved your CAPAs, and over the next few years, they will continue to monitor, support, and work alongside you to verify implementation, effectiveness, and regulatory compliance. I'd like to take a second now to welcome Dr. Ronnie Bolag, who will be elaborating on some of the common biological product deviations that you will that you will be encountering. Dr. Boyd?
Thank you very much. I would like to just explain some of the ongoing reporting processes that are important for provide for the FDA, and this has to be done in real time, and it should happen before the inspection. Biological product deviations, or anything that deviates from your protocol and that may potentially affect the safety, purity, or potency of your product. These need to be submitted as soon as possible, but no later than 45 calendar days from the date the information was received. And for fatalities, which is any complication of transfusion that led to a fatality, that it needs to be investigated, documented, and reported, and that has to be done within 7 days of the fatality or of the fatality coming to light. So I was going to give an example of one such incident. This is a case report. This was quite a number of years ago. An 87-year-old African-American male brought by ambulance from the nursing home for respiratory compromise. He had diastolic heart failure, diabetes, hypertension, and he was symptomatically anemic. On chest x-ray, he had bilateral lower lobe airspace opacities and a left-sided pleural effusion. His labs showed a slightly elevated white count and a slightly decreased hemoglobin. So he was started on antibiotics, and they gave him, per protocol, 2 units of red cells for his symptomatic anemia. And within 4 to 5 hours, the patient was being watched, and the team showed that he was experiencing tachycardia and hypotension, and he was desatting to below 75%, and they determined that he required intubation. He progressively deteriorated, and on the next day he experienced ventricular tachycardia followed by bradycardia and PEA. A code was called at that time and several times over the next 2 days. He really deteriorated and continued hemodynamic instability, developed AFib and acute renal failure. After 5 days, the patient's family decided to withdraw care, including the extubation, and the patient expired. So we were notified after this period that they suspected a trolley may have contributed, and so we did a transfusion reaction assessment, and we determined that the patient did in fact likely experience trolley transfusion-related acute lung injury after the transfusion of those 2 red cells. And that's based on his dyspnea and hypotension within 6 hours of the transfusion, and the fact that chest x-rays showed bilateral pulmonary infiltrates. We sent segments from both RBC units for HLA testing, for antibody testing, and one unit had class 1 HLA antibodies of about 20%, and the second unit had both HLA class 1 and class 2 antibodies at a fairly pronounced level. This was consistent then with trolley. So we subsequently notified the FDA as soon as we successfully concluded that this was a transfusion-related fatality. We sent them the death certificate, the transfusion reaction forms. They asked for compiled lab values and microbiology culture reports. His pneumonia was due to Klebsiella pneumonia infection. The notification center, the collection center was notified that they had these 2 donors that have antibodies to HLA and should be excluded from donating plasma products. We shared all this information at the next FDA inspection, and the case was reviewed, and the FDA representative determined that all of our actions were appropriate. So huge sigh of relief. This is to kind of emphasize that there are fatalities associated with transfusion, and this is a compilation of 5 years worth of data from the FDA. These are all reported events. And you can see that there were about 30 to 40 events per year, and about 20% of those were Crawley. So fatalities hopefully will not be a common occurrence in your lab. However, there are any kind of biologic product deviation should be reported to the FDA. And I just compiled two cases that were reported in the literature as examples. The first case was phlebotomist initials missing from the label. A unit was cross-matched and transfused, and the information was not discovered until the next day. Phlebotomist was off, and his initials were then added on the 4th day. But this should this be reported? And the answer is yes. This should be a product BPD with a code, with a specific code saying that routine testing, sample identification, and was incorrect or incomplete. Another potential case that could happen on any given day. This is the blood bank. The blood bank ID on a unit of red cells differed from the tag by 1 letter, and it was dispensed, but subsequently returned by the floor to the blood bank for clarification, and so was not transfused. But this should be reported as a biological product deviation because of an incorrect cross-match tag or tie tag or missing information, unit, lot, or pool number, pool numbers. And these just serve as actually 3 common cases of reportable events. And I'll turn it over now back to Dr. Okariz to complete our presentation.
Thank you, Dr. Bullock. So now the question becomes, how do you build a system that's continuously regulatory compliant? Excuse me. You want to make regulatory compliance a natural result of your purpose-built systems designed to provide quality patient care and safety. Understand that quality work and outcomes are the result of a workplace that values that quality, and these values need to be promoted through the leadership and the quality management team. Most systems work well and as designed until their components are sufficiently stressed. When this happens, latent system errors reveal themselves. It's up to you to determine the cause of those systemic failures and find appropriate solutions for them. Root cause analysis tools allow you to thoroughly analyze an error. A fishbone diagram, for example, can be used to map out environmental, equipment, and personnel factors for complex issues. And the 5 whys method forces you to dig past the surface of an error to find the true root cause. Generally, the true cause of an error will fall under one of 2 categories, a systemic failure or a personal failure. The remainder of this presentation is dedicated to providing you with effective strategies for mitigating both. The systems approach states that most errors occur due to poor system design rather than individual failure. Use the substitution rule to determine if the error is caused by a broader systemic issue. Could anyone else have made the same error, or is there a single staff member repeating the same error? The allocation of resources and personnel dedicated to your quality system is crucial. An efficacious quality management team is your most powerful tool for assessing and improving your systems. Their role is to develop and review new policies and SOPs, keep your systems updated on the latest regulatory changes, and ensure internal regulatory compliance. As an extension of your quality management team, a multidisciplinary quality management committee will ensure that proposed changes are reviewed and verified by individuals from various perspectives at all levels of patient care at your facility. When developing your policies and SOPs, standardize your processes and eliminate competing methodologies to reduce confusion. Perform bench level validation by giving new SOPs to bench techs to read cold. If the person doing the work can't follow it, the document is essentially useless. Implement the use of software solutions to manage your documents. Electronic record services such as Media Lab's Inspection Proof is useful for real-time tracking of CAP checklists and documentation. Likewise, Master Control is great for managing document manufacturing, CAPA development, and incident tracking. Reduce operator error by implementing an automated vigilance system using computers to verify patients and products. Constrain the available options so operators are forced to pick the correct one. For example, if clinicians are continuously forgetting to put an order for post 1 hour platelet count, after platelet transfusion, combine the orders in your EMR and make it impossible to forget. Keep it simple, stupid. A 50-step process has an estimated 39% chance of error if each step only has a single 1% risk. Make your systems and processes as simple as possible. Life is unpredictable, and even the best laid plans will inevitably fail. Be proactive and anticipate the potential latent errors in your system. Design your systems so that they encourage error detection and correction before the problems occur. A healthy workplace culture that encourages personal accountability is vital to a quality system, but is frequently overlooked. Ensure your staff are familiar with your policies and SOPs. There should be no guesswork regarding their responsibilities. Then validate their performance and competency to ensure safety and quality standards. Encourage strict adherence to the established SOP. This assures the staff that errors are the result of a system fault and not personal skill or their abilities. Ensure your staffing requirements are met and sufficient to accommodate unexpected events and absences. Provide your staff with the environment and the tools required to perform their tasks correctly and efficiently. Adopt an open door policy that makes leadership approachable. The staff should feel comfortable calling for help to avoid making a mistake. Additionally, teams function better when communication is not constrained by rank or job titles, so reduce hierarchical barriers in the workplace. Foster a work culture, a just culture that prioritizes fixing the system rather than punishing the person, and promote a non-punitive environment. Blame and shame are destructive and lead to self-protective behaviors that hinder error reduction. Hierarchy and aggression stifle honest communication. Therefore, leadership must be attentive and address toxic individuals who jeopardize team cohesion. All in all, a healthy workplace environment with quality system that works as intended should see an increase in reporting. This is a sure sign of a healthy culture where staff feel safe in self-reporting mistakes. Communication is another key component to a quality system. Here are some strategies for improving it system-wide. Handoffs are focal points for error. Simplifying the process and reducing their frequency decreases error rates. When handoffs do happen, perform clipboard audits, where intentional observation of the processes, such as blood release to the OR, ensure that interdepartmental handoffs are following safety protocols. Human memory is fallible, so provide memory aids such as checklists and protocols in easily accessible and strategically placed locations. Provide your staff with easy and convenient access to important information such as readily available electronic medical record system. And use standardized communication tools such as SBAR or situation, background assessment and recommendation, and closed loop communications or readbacks to improve understanding and comprehension. Gemba boards are a great way to show staff that their feedback is being tracked and addressed. It builds trust between departments by displaying real-time performance, progress, and issues in the workplace. And lastly, the use of networking as a resource. A face-to-face networking at professional meetings such as AABB, CAP, and the GAP is a great way to pick the brains of other directors and solve shared institutional problems, gain insight and advice from experts on the field, collaborate and foster ideas and innovations, and improve communication skills and build valuable relationships. Performing regular mock inspections is absolutely critical for being inspection ready. Mock inspections highlight your facility's weakness and allow you to address them ahead of regulatory organizations. Involve your staff in the self-inspection by familiarizing them with regulations relevant to their post. Then have them inspect and evaluate their workplace, workflow, and compliance to SOP. This will increase their knowledge of regulatory requirements, demystifying the inspection process. By promoting quality and honest reporting, your staff will serve as a trusted partner in the inspection process. They can even provide you with bench-level insight and perspective your team may not have previously considered. Design your systems to be prepared for anything and everything. Have multiple layers of backup plans that have been validated to meet safety and quality standards. Maintain physical downtime binders for when electronic systems fail, and establish triage protocols that prioritize stat specimens during manual processing. And conduct mock downtime drills so that your staff are not panicked during real-time system failures. And a final point about the FDA. As scary as they seem, the FDA does not want to shut you down. They want you to provide patient care with at least a bare minimum standard for quality and patient safety. They are an extra set of eyes observing your systems, helping you identify weaknesses, and working with you to correct them. Treat them as part of your quality team and foster a trusting relationship with them. They'll keep you on the right path. Thank you. And a special thank you again to everybody who has contributed to this project. Again, I don't think that this would have come out nearly as good as it did without all of your help. We do have the actual presentation tomorrow. Unfortunately, that's not being recorded. But if any of you have any feedback that you think may help us in our presentation tomorrow, that would greatly be appreciated. You could